We are continuing on the tools and techniques of biotechnology. Specifically for All India Medical, it's important that we know some more points in the context of CBSE. Well, we are again coming back to bioreactors, and we need to investigate what are the advantages of bioreactors. The advantages of bioreactors when compared to flask cultures or flask stirred cultures. So, when you take bioreactors, first very important point is they provide continuous aeration. Next, there is thorough mixing of the products. There is thorough mixing of the products. The products are stirred. Third important advantage of a bioreactor over a flask stirred culture is there is large scale production. When you say large scale production, there is large scale production of antibiotics, vaccines, hormones, enzymes, thermopeuticals, etc. Large scale production, that is for mass scale. On a mass scale. In a bioreactor, it maintains an optimum growth condition of the microbe. Optimum, it is not maximum or minimum, but efficient. When you say optimum, it means efficient growth. It maintains optimum growth conditions, so that there is increase in cell number and also increase in cell size, and the microbial cells will go through the sort of an exponential phase. If you recollect microbes, primarily when you take microbes, they go through a log phase, then they go through a plateau, and then they fall down. So, this is termed as a sort of an exponential phase when the microbial cells increase in cell number and also in cell size. So, the exponential phase of the growth is primarily maintained by efficient growth conditions. By efficient growth conditions, that is, the growth conditions in a bioreactor are regulated. It is regulated by periodic inputs and outputs. You periodically regulate the temperature, you regulate the pH, you regulate the substrate, as well as even the vitamins or the hormones or the nutrients which are added, including oxygen. Thereby, what exactly gets regulated in a sort of a bioreactor is we have temperature control, then we have a pH control, oxygen control is over there. Then again we also control the substrate input and outputs, then also additional vitamins and as well as salts. So, all of these are primarily regulated in a sort of a bioreactor when compared to a flask stirred culture. Thereby, we understand that if you take a bioreactor, there are several sort of an advantages. The advantages is we facilitate the cell multiplication. Now, the higher the cell multiplication, it means it leads to efficiency. We are talking about efficient cell multiplication. The reason why there is efficient cell multiplication is every time in a sort of a bioreactor, if you recollect a sort of a bioreactor, every time in a bioreactor we have fresh medium added. There is a fresh medium added, and periodically the medium is also drained. Thereby the cells can take in the medium and show high physiological activity, or they can show high metabolic activity. When fresh medium is added, thereby the cells are metabolically active. The cells are metabolically active. Once the cells are metabolically active or physiologically active because the medium is being replaced, all the cells will be getting into the sort of a log phase. This is the log phase or the logarithmic phase. The cells would be in the logarithmic phase of growth, and once the cells are in the logarithmic phase of growth, they always have larger biomass. So the biomass increases, the total amount of productivity or the total amount of chlorophyll or the biomass increases. This again leads to high yield. It leads to high yield, and we get the sort of a desired protein. Thereby we understand logarithmic growth of the sort of a cells in a sort of a continuous culture, because primarily how is it maintained in a sort of a continuous culture? It is maintained in a continuous culture by means of an aerator. We have an aerator which again is agitated, so you call it as agitator. It agitates, and because there is an aerator and an agitator, there is high amount of oxygen delivery. There is high amount of oxygen delivery. Simultaneously, we also have temperature and pH control. So oxygen delivery, temperature and pH control are very important in a sort of a bioreactor. Bioreactors also have periodically they have a sampling port. If you recollect the diagram, they always have a sort of a sampling port from which the nutrients as well as the samples are drawn. So there is a periodic withdrawal of the samples to test in which phase of the growth are the sort of a cells. And if you look at the bioreactor, because it is primarily a method of growing cells under aerobic as well as anaerobic conditions, we also can call a sort of a bioreactor as a sort of a biofermenter. It is popularly called as a sort of a biofermenter. So these were the sort of an advantages when compared to the flash shaped stirrer, there is aeration, mixing, large scale production, optimum growth, regulated outputs, larger biomass, sampling port, oxygen delivery, cells are metabolically active, fresh medium is added, it shows the exponential growth of phase, cell multiplication leading to higher efficiency. So these are the sort of advantages of a sort of a bioreactor over a sort of a flask culture.